All right, guys, so we're going to pick up where we left off. We've been discussing the three major domains of taxa, the bacteria, the archaea, and the eukaryotes. What we're going to talk about is a couple major themes for all of these different groups of organisms. Number one, how do they respond to their environment? Number two, how do different taxa maintain homeostasis? Number three, how do different taxa reproduce? And number four, how do different taxa acquire and process nutrients? And so we've discussed this idea of responding to the environment. We've begun discussing how different taxa maintain homeostasis, and this is where we're going to be for the next couple of lectures. So remember that homeostasis is when organisms actively regulate and maintain a constant internal state in the face of changing environmental conditions. So homeostasis is all about maintaining balance, and we talked about a lot of different factors that organisms have to maintain homeostasis in, or create a balance for within their bodies. And two of the major factors that we're going to explore in the next couple of lectures are water and electrolyte balance, something called osmoregulation. And we're also going to talk about temperature regulation or thermoregulation. So these are two important factors that organisms have to maintain homeostasis for, or have to maintain a balance for, to survive. So let's begin talking about water balance or osmoregulation. So when we talk about osmoregulation, we're going to spend the next couple of lectures talking about three sort of major groups of organisms and how they osmoregulate. We'll talk about unicellular taxa, we'll talk about some simple multicellular taxa, and we'll talk about complex multicellular taxa. We're going to be talking about each of these groups in 1, 2, 3 major types of habitats: in fresh water, in salt water, and on land. And that's specifically because these different groups of organisms have evolved different adaptations to deal with the different sorts of pressures that these different habitats present. So we're going to talk about the adaptations these groups have evolved that enable them to osmoregulate. But before we get into osmoregulation, we have to have a review of osmosis. And if all of this stuff that I'm talking about in the next few slides looks completely new to you, please make sure you first view that osmosis review lecture and quiz that's posted on Canvas. That's going to help you a lot with this information because I'm not getting into some of the initial stuff on this. So remember or recall from that previous lecture that osmosis is diffusion of water. And basically, it's going to determine the water and electrolyte content of cells. So for example, if you can think of this sort of flask here that has a split in half, and somewhere in that split in half, there's an artificial lipid bilayer. And remember that a plasma membrane or a lipid bilayer is what a cell membrane is made up of for organismal cells. And you can imagine in this liquid, you've got these green things here that represent dissolved ions or a solute that's dissolved in the solution. And assume this solute or those ions cannot readily pass through this lipid bilayer. So a lipid bilayer, it's selectively permeable. It allows some things to pass through it, other things can't. So let's pretend that these green ions here cannot pass through this lipid bilayer. Which side will water move to by osmosis, side A or side B? Okay, if you said the water will have a net movement to the right side, side B, you are correct. And the whole idea here is the water is trying to balance out the concentration of solution on the two sides so that it's equally dilute on both sides. So given that, how will osmosis work in salt versus fresh water? So in other words, of choices A, B, and C, which represents osmosis in salt water? So which of these best represents the net direction or the net movement of water in salt water solution? Okay, if you said C, you are correct. So remember these green balls here, those represent salts or ions that are dissolved in the water. And so salt water is typically hypertonic relative to the cell. So hypertonic means there's a higher concentration of salts outside of the cell than there are inside the cell. And so there's going to be, in general, a net flow of water out of the cell to basically balance the concentration so that the concentration inside the cell of solutes matches the concentration of solutes outside the cell. Okay, how will osmosis work in fresh water? In other words, which of these diagrams, A, B, or C, represents the net movement of water into, out of a cell in a freshwater environment. And remember, this here represents the cell with a lipid bilayer around it. Okay, if you said A, you are correct. So remember that freshwater is typically hypotonic relative to the cell. So hypo, it means less. And what you can see is there's a lower concentration of salts in that freshwater environment than there are inside the cell. And so there's going to be a net movement of water by osmosis into that cell to make the concentration of ions in the cell match the concentrations of ions outside the cell. And remember, the key assumption here is that these ions are not able to move in and out of the cell. Only the water can make that concentration of ions balance out. So there'll be a net flow of water into the cell in a freshwater environment. Okay, so given that, which of the following best represents how an animal cell might behave in a saltwater solution? So A, it's going to blow up. B, it's going to remain about the same size. Or C, it's going to shrink. Okay, if you said C, you are correct. So salt water, remember, it's typically hypertonic. And so there's going to be net movement of water out of the cell so that there's a higher concentration, or the concentration in the cell matches the concentration outside of the cell. So the net flow of water outside of the cell means that that cell's going to have, or it's going to shrink because water is going to move out of it. And this is often referred to as something called crenation. And that's just a fancy word for a cell sort of imploding on itself because all the water is getting sucked out of it. And as you can imagine, crenation is typically a bad thing for a cell. Okay, next question. Which best represents how an animal cell might behave in fresh water? So how might an animal cell behave in fresh water? A, B, or C? All right, if you said A, you are correct. So remember that fresh water is typically hypotonic relative to the cell. So remember, there's a lower concentration of solutes outside of the cell than there are inside of the cell. Cells tend to keep a good amount of salts inside of them. And so there's going to be a net flow of water into the cell. And sometimes if too much water flows into the cell will fill up too much with water and the cell membrane will actually explode. And so this is often referred to as cell lysis, which is basically just a fancy word for the cell being broken down or exploding. And as you can imagine, this is a bad thing to have happen to cells as well. You don't want cells to lyse in general if that cell's going to survive. Okay, so given that background information, let's think about how these different sort of groups of organisms can survive in these different habitats and osmoregulate or balance the amount of salts and electrolytes and water that they have in them so that they don't explode or implode. And let's first look at this groups of unicellular taxa. So we'll look at protists and we'll look at prokaryotes. Remember, protists are eukaryotic unicellular organisms. And we'll look at them in a freshwater environment. And remember, a freshwater environment is an environment that does not have a high concentration of salts in it. Okay, so just to remind you here, freshwater is typically hypotonic. So in other words, there's a lower concentration of solutes in the freshwater relative to what's in the cell. And that means that the pressure that unicellular organisms are dealing with in a freshwater environment is potentially too much water rushing into the cell and the potential for the cell to explode. So because there's this net flow of water into the cell, cells need to figure out ways to stop from bursting or to stop this net flow of water into the cell or reduce that net flow of water into the cell if they can. So let's look at some of the adaptations organisms have to do that. So how do organisms, unicellular organisms, deal with that? So one key adaptation that unicellular species can use in fresh water to keep from popping is a cell wall. So both prokaryotes and unicellular algae, and remember prokaryotes, these are bacteria and archaea, whereas algae are eukaryotic organisms. Remember, there's a difference between prokaryotes and eukaryotes. But both prokaryotes and eukaryotes, some eukaryotes, can have a cell wall. And that cell wall is basically just a polysaccharide matrix that's outside the plasma membrane that resists expansion. And so basically, this is like a strong sort of net-like series of molecules that will hold the cell together and stop it popping. It's like basically adding, you can think about it as being like Spanx that add this extra layer of protection that stop the cell from expanding too much. It's like a pair of pantyhose around it, but even stronger than that. And it's important to make a distinction that a cell wall and a plasma membrane are two different things. So that lipid bilayer that we've been talking about that make a cell membrane, That is this plasma membrane. The cell wall is something different that's beyond, it's a level of protection beyond that plasma membrane or that lipid bilayer that we've been talking about previously. So unicellular eukaryotes that don't have cell walls, many of them can have something called a contractile vacuole to stop the cell from popping, from all the water from rushing in a freshwater environment. And basically what a contractile vacuole is are compartments that take up excess water inside the cell and contract to expel it out. So it's basically like a release valve that shoots that extra water as it comes in out so that the cell doesn't expand too much. And so you can see here, this is a unicellular eukaryotic organism. It's also called a protist. And this protist, it's called paramecium, has two contractile vacuoles right here. And you can see right here, this is just a video of those contractile vacuoles. It kind of looks like a set of eyes winking at you. Those contractile vacuoles, once again, water is rushing into this cell because it's saltier than its environment. And that contractile vacuole basically sort of winks closed to push that water out so that the cell doesn't expand so much that it pops. Okay, so two examples of adaptations that unicellular taxa have to stop from popping in a freshwater environment. Contractile vacuoles are one example that are unique to eukaryotes. Cell walls are one example that both prokaryotes and some eukaryotes have. So those are two examples of adaptations. Now let's look at adaptations that organisms have, these unicellular organisms have to survive in salt water. So remember that salt water is typically hypertonic relative to the cell. So there's more salts in the environment than there are within the cell. Thus, there's a net flow of water out of the cell. So the cell can potentially shrink or crenate. And that shrinking can eventually lead to cell death. So this can be very bad. And it's very tough for organisms, many organisms, unicellular organisms, to survive in very salty environments. And in fact, we've used that to our advantage. And that's one reason why curing meats works really well. So most disease-causing bacteria won't grow if salt concentrations are greater than 10%. So if you can greatly increase the salt concentration around something that would typically rot, you can actually kill all the bacteria because they can't osmoregulate. All the water gets sucked out of them and they can't survive. And so curing meats with salts is a great way to preserve them into the long term and get all the water out and stop bacteria from growing. So given that salts can be really effective in killing off smaller organisms and unicellular organisms, how do unicellular organisms deal with salts? So bacteria, lots of unicellular organisms can survive in salty environments and salt water. So the ones that can survive, how do they do that? So one adaptation that unicellular organisms can use to keep from shrinking in salt water are ion channels, okay? So if we're looking at a plasma membrane here, so this is that lipid bilayer. You can see the cytosol here. So this is the inside of the cell, and this is the extracellular fluid. So this is the outside environment outside of the cell. And these ion channels are these purple things here. And essentially what these are just proteins that are embedded in the cell membrane. And those ion channels can be permeable to solutes. So remember, ions, these charged particles, so things like salts and things like that, that are dissolved in salt water, can't readily pass through a lipid bilayer. They can't pass through the cell membrane. It's impermeable to ions. However, these ion channels can create special gaps that allow these ions, things like calcium, potassium, sodium, to pass through the cell membrane and go into or out of the cell. And so you can think of these ion channels sort of like the bouncers of the club that determine who can get in and who can get out. And they can selectively open and close to sort of regulate the movement of ions into and out of cells. And so ideally what an ion channel will do is allow some solutes to move in so that you get an isotonic environment or a balanced environment, an equal amount of solutes inside and outside of the cell so that the cell's not shrinking or popping, okay? So given that, which direction would you predict that ion channels are gonna pump in salt water to maintain osmotic balance? So if you have a cell that's in salt water like this and it has ion channels, which direction is it going to pump the ions? Into the cell or out of the cell? Okay, so if you said into the cell, you are correct. So remember, in this situation, there's a net movement of water out of the cell. So if you want to stop water from moving out, You got to pump ions in so that you have an equal concentration of ions on either side. And if you can get that isotonic environment so that it's a matched concentration of ions inside and outside of the cell, the water will not have a net movement out and the cell will not shrink too much. And in general, you can just think of the fact that water is going to follow the directions that you pump ions. Okay. So which direction would you predict channels are going to pump ions in a freshwater environment to maintain osmotic balance in a unicellular organism? If you said out of the cell, that is correct. So remember, in a freshwater environment, there's typically a higher concentration of solutes inside the cell, and so there's a net movement of water into the cell. So you're going to want ions to pump, ion channels to pump ions out of the cell so that you get an isotonic environment and there's not too many ions inside the cell so that too much water doesn't go in and pop the cell. And an important thing to make note of is that animal cells typically require an isotonic environment to survive. Okay, so for animal cells, you've either got to have the environment matching the typical salt concentrations that are inside the cell so that there's a net movement, neither a net movement into or out of the cell so that it won't pop. And so isotonic, remember, is the same concentration of solutes inside and outside of the cells. And that's because animal cells don't have cell walls and they don't have contractile vacuoles. So they don't have a lot of ways to deal with different ion concentrations. And this is really important, for example, in human red blood cells, where in a hypertonic environment, human red blood cells will shrink or crenate and implode. In a hypotonic environment, they'll explode really easily. And so they really require an isotonic environment to keep from dying. And so this is really important for maintaining the water levels in your bloodstream, and that's why one of the major reasons that it's so important to stay hydrated and maintain electrolyte balance, because your red blood cells are just like Goldilocks. They need their concentrations just right to be happy. And this is why, if you've ever had an IV bag before, they always have a 0.9% sodium chloride solution. So it's hugely important that they have just the right concentration of salts, because if they put the wrong concentration of salts in an IV bag, what that's going to do is create an either hypertonic or hypotonic environment that's going to pop all your red blood cells. So those IV bags, just like Goldilocks bowl of cereal, needs to be just the right temperature. The IV bags have to be just right. Otherwise, your red blood cells are going to pop. Okay, so just to sort of recap, we've talked about unicellular taxa and some of the adaptations that they have to survive in freshwater. We're also going to talk about these other adaptations, which are ion channels that both prokaryotes and eukaryotes have, and they can help them osmoregulate in both a freshwater environment and in a saltwater environment. So ion channels, very important. So the big sort of take home here is that organisms have evolved adaptations to regulate diffusion and osmosis so that they can maintain cell size, and those unicellular organisms don't have their cells exploding or imploding. I also want to make this important note of this important caveat that this doesn't always mean that the cell environment and environment have to be isotonic. So organisms are constantly doing this sort of pumping of water in and out or ions in and out. And actually rarely will you find, or oftentimes you won't find, an individual organism matching its environment in terms of tonicity. It's got to be constantly sort of pumping things in and out to be able to stop the cell from imploding or exploding. Okay, true or false? Most unicellular organisms have adaptations to help them maintain osmotic balance. That is true. And we just talked about some of those. Next question. Most unicellular organisms need to be in an isotonic environment to maintain osmotic balance. This is actually a false statement. So many unicellular organisms can be in a saltier or more dilute solution, so a freshwater environment or saltwater environment, and are still able to maintain osmotic balance or stop from exploding or imploding. It just means that they have to do a lot more work to do that. So they do not have to be in an isotonic environment to survive. It just means when they're not in an isotonic environment, oftentimes they have to do a lot more work of pumping things in and out to survive. And number 3, most unicellular organisms cannot survive if they're not in an isotonic environment. This is false. Most unicellular organisms can survive when they're not in an isotonic environment. And number 4, prokaryotes use contractile vacuoles, cell walls, and ion channels to osmoregulate. True or false? This is false. Contractile vacuoles are unique to the eukaryotes. They're sort of like a type of organelle. And remember that prokaryotes do not have organelles. So that's something that's unique to eukaryotic organisms. Okay, so we're going to end there for now. We'll pick up on the next lecture talking about simple multicellular taxa. Thanks, guys.